Okay, children. So now we will be starting the next topic for the discussion. And yesterday we discussed about the discovery of the cell, who discovered the cell, who discovered the first living cell, then who discovered the nucleus, who gave the term protoplasm, then the cell theory points we have discussed, and microscope structure and the function of it that we have discussed yesterday. Now moving to the next topic, a very important topic, which is the difference between the prokaryotic and the eukaryotic cells. So prokaryotic and eukaryotic cells. When we talk about these two names, prokaryotic. So it is like from the two names we are getting the word prokaryotic, and this karyotic word is basically derived from the word called as karyon, which means the nucleus. So karyon means the nucleus, and pro means old, or we can say as primitive, or it is also called as the false. So when you join these two words together, which is like old, primitive, and this word karyon, nucleus, it gives you the old nucleus. Or we can say it as the primitive nucleus. So those cells which has the old nucleus or primitive nucleus or the false nucleus, such cells are called as the prokaryotic cells. Now here, the old or the false means it looks like the nucleus, but actually it is not the nucleus. In this prokaryotic, the nucleus is not clearly visible at all. So that is, those cells are called as the prokaryotic cells. Fine, Ketra, Hansini, Priyamvada, Sahiti, Shrivatsa, and Yashoda.
Ma'am, do we write this?
No need to write. You have already written.
Ma'am?
Yeah, tell me, Yashoda.
Ma'am, can you once show the screen again? Sorry.
Yes, I'm showing, no problem.
Hello,
So
ma'am.
it has been derived. Yes, ma'am.
Ma'am, if you're not allowing kids to write, right? But at least you show, ma'am. That will save time also, and that will visually they will see the picture of it. You are writing anyway very fast.
Yes, ma'am.
If
Yes,
they
ma'am.
can
Yes, ma'am. I'll be.
and you can write and make them read one of them. That will help them remember. It will take probably
Sure,
a
sure.
minute or. Thank you.
So, welcome. So, prokaryotic derived from the two words, which is like basically pro means old, primitive, or false, and karyotic means the nucleus. So, the complete word means the old nucleus or the primitive nucleus, which means they have the nucleus, but that nucleus is not fully developed or the completely developed. Such cells are called as the prokaryotic cells. Yashoda, have you written that part?
Ma'am, 1 second. I'm almost done.
Okay. Srivastava, can you tell me what is prokaryotic?
Prokaryotic is like
Yeah, tell me.
Prokaryotic.
What do you mean by pro?
Pro is like professional.
No. Old, primitive, false. Any one word you can use for it, okay? Pro means old, or you can say it is primitive, or you can say it is false. And karyotic is basically karyon, which means the nucleus.
I'm done.
Okay? So those organisms, okay, Yashoda. So those organisms are made up of the old nucleus. They are called as the prokaryotic cells. Now coming to the second part of it are the eukaryotic cells. Eukaryotic cells. Now, in this also, what happened? Two words are there, eu and karyotic. And I told you, what is karyotic? Karyotic is a word derived from the karyon. Srivasa, what do you mean by karyon? Yes, Srivasa, what do you mean by karyon?
Did you ask me? Oh.
Yes.
Carrier means like a advanced, I can say.
No, karyon means nucleus. Okay.
That's it. Yeah, nucleus.
Karyon means nucleus. So in the eukaryotic, what happened? The cells are basically, the nucleus are basically advanced, new, or true. So any one word you can use it. In the eukaryotic organism, the cells will have the advanced nucleus, or we can say they have the clear nucleus, or we can say they have a well-defined nucleus. So if in any organism, if they have the well-defined nucleus or clear nucleus is present to them, then they are called as a eukaryotic organism. So prokaryotic, in them the nucleus will not be visible at all, whereas in the eukaryotic the nucleus will be visible. So that is what the eukaryotic and prokaryotic. Now one by one we will be discussing the differences between the prokaryotic and eukaryotic cells. So this is very, very important for your exam, the differences between the prokaryotic and eukaryotic. Yashoda, have you done? Did you write it?
Yes, ma'am.
Do you have any doubts, Leah?
No, ma'am.
Okay. Now going to the next one is the differences between the prokaryotic and eukaryotic cells. The first difference, as I told you, prokaryotic nucleus is absent, whereas in the eukaryotic, the nucleus is present. Second difference, in the prokaryotic, now if the nucleus is absent, the nuclear membrane is also absent. Nuclear membrane is also absent. Whereas in the eukaryotic, the nuclear membrane is present. Then the next one, in the prokaryotic, they contain single chromosome. So whosoever the prokaryotic organism are there, like bacteria are there, some algae are there, whosoever are prokaryotic, what do they have? They contain only single chromosome. They will have only one chromosome in them. Whereas in the eukaryotic, they have...
Single chromosome, is it like an entangled mass of like
Correct, exactly.
stuff?
Yes. Whereas in the eukaryotic, they will have the more than one chromosome present in them. Then in the prokaryotic, they do not have membrane-bound organelles. It means the organelles which has outer covering, that is called as membrane-bound. So they do not have outer layer of the covering in some of the organelles. So membrane-bound organelles are absent. They do not have it. Whereas in the eukaryotic, membrane-bound cell organelles are present. They will have this cell organelles present in them. So these are the 4 points. Still, we should write 2 more points. Let me complete those points and then I'll be coming back again to this page. Then in the prokaryotic, the cell division takes place by fission. So this is like method. The method by which they divide, that is called as fission. Fission means division. And the second method is called as the budding, budding. Whereas in the eukaryotic, the cell division will occur by either meiotic or by mitotic division. So two type of the division will happen in them, meiotic or the mitotic division. Meiotic means whatever the chromosome numbers are there, they will reduce half, and mitotic means the number of chromosome will divide into two and the number of chromosome will remain same in them. Then the last point, the size of this prokaryotic cell is generally small. They are small in size, whereas in the eukaryotic, the size of a cell is large. Generally, they are large in nature. So these are the differences between prokaryotic and eukaryotic. In the prokaryotic, nucleus is absent. Eukaryotic, it is present. Prokaryotic nuclear membrane is absent. In the eukaryotic, it is present. In prokaryotic, they will have single chromosome. In eukaryotic, they will have more than one chromosome. In prokaryotic, the membrane-bound cell organelles are absent, whereas in eukaryotic, they are present. Then size of prokaryotic, it is small. In size of eukaryotic, it is big. Then the cell division in the prokaryotic, it will be taking place by the fission or budding. And cell division in the eukaryotic, it will be done by the meiotic and the mitotic division. Kirtana, am I clear with this? Yes, Kirtana? Sahiti, am I clear?
Yes, ma'am.
Okay. Kirtana, Yashoda? Yashoda done? Should I
Yes,
explain
ma'am.
anything again?
No, ma'am, I'm good.
Okay, good. Srivatsa?
Uh,
Yes. Read this.
yes. Prokaryotic, right?
Yes, yes. Prokaryotic, you can read.
So nucleus is absent. Nuclear
Hmm.
membrane also absent.
Very good.
Not contains, they contain only single chromosome.
Correct. Then,
Membrane, membrane bound organelles are absent.
then,
Cell division takes place, fusion or budding.
correct. Then,
Size of a cell is generally small.
okay. Now, can you tell me without seeing the opposite of that? If the size of the prokaryotic is small, then in the eukaryotic, what will be the size? Small or large?
No, it will be large.
Correct. Okay. Then, if the nucleus is absent in prokaryotic, so in the eukaryotic, the nucleus will be present or absent?
It will be present.
Very good. Okay. So, whatever is absent in the prokaryotic, same way in the eukaryotic, it will be present.
Present.
Clear?
Yes, clear, ma'am. Now clear.
Okay. Very good. Very good. Shall we go to the next topic? Ketna, Sahiti, Priyamvada, Srivatsa, Yashoda, Hansini.
Yes, let's go.
Okay. So, yes. Now, again, a very important part. In the 3 marks, they will be asking you to explain the prokaryotic cell with the diagram. Okay. Every year in the school, they will be asking this for the 3 marks question that you need to explain the you need to explain the prokaryotic cell. Now, when we talk about the prokaryotic cell, bacteria cell is an example of that. So, bacterial cell is what? It is a prokaryotic cell. So, this is a diagram which you can see over the screen. One by one, we are discussing the parts of it and also the functions of the part. Now, starting with this hair-like structure. This hair-like structure, which you can see all over the bacteria, these are called as pilus. They are called as pilus. Now, this pilus, what are they? They are the thin hair-like structure that will be coming out from the cell surface of the many bacteria. The main function of this hair-like structure called as pilus is that they will be attaching themselves to the surface. So wherever they go, whatever surface they go, they will attach to that surface with the help of this hair-like structure called pilus only. So if they have to attach to the surface, they will use this pilus. And if they have to attach themselves to the other bacteria, again, they will be using this pilus only. So pilus is basically like, the Fevicol. If you put the Fevicol and you stick something to it, what happened? They will get joined together, combined together. Same way, the pilus are here like structure, with the help of which, wherever they want to go and attach themselves, they will be attaching to that surface, particular surface or the organism also. That is what the pilus will be doing it. The next structure is a capsule, which is this part, the outer part, this part is called as a capsule. So capsule is an outer layer that protects the bacteria. So the bacteria will be protected because of this outer protection called as a capsule. This will be helping the bacteria to attach themselves to the surface or to the other organism, and also they will be acting as a kind of the defense mechanism. So if they want to protect themselves, what they should do? They should fight, right? So because of this capsule only, they will be fighting. Now, for example, if you are not feeling well, what your mother will be doing? Either they will be giving you some kada or they will be giving you that medicine, right? So that kada or that medicine, what they are doing, they are protecting your body, correct? So capsule also behave in the same way where they will be getting the or they will be giving the protection to the bacteria. Am I clear, Srivatsa? Hansini, Sahiti, Yashoda, Kirtana?
Okay.
Yes, ma'am.
Okay. Then the next one, cell wall. As we know from, I think, from past 2 years, we are learning about the cell wall. Only in the plant cell, we have the cell wall. So bacteria also have the cell wall. So what do they do? Whatever shape you can see, this shape, whatever the oval shape or other shape also, there are many different shapes of the bacteria are there. So whatever shapes are there for the bacteria, that will be given by the cell wall. So cell wall main purpose is to maintain the shape of the bacteria and to give the protection to them. That is what the bacteria will be doing. The next one, plasma membrane. This is internal membrane, this one. So they will be regulating the movement of substances in and out of the cell. So whatever the things bacteria need, they will be taking it. And whatever they do not need it, they will be giving it out. For example, now if the structure is like this, okay, suppose this is the bacteria and here this is we have the cell membrane. Now what happened? They do not need everything. Some things only they require. Might be, for example, if they need the oxygen, what do they do? Through this cell membrane, they will be taking this oxygen inside. And then they do not need the carbon dioxide. So what do they do? Again, from this same layer, they will be removing that carbon dioxide outside the body. So this is what the entry and exit of the material will be done by the cell membrane. So cell wall will be giving shape to the bacteria, whereas cell membrane will be allowing for the entry and the exit of the substances in and out. Then the next structure we have is the flagellum. This you can see, the tail-like structure. These are called as flagellum. They are called as flagellum. Now this flagellum, what do they do? They help in the movement. Like how do we have the legs? With the help of the legs, we move, right? Same way, the bacteria, they also have to move from one place to other place. They do not have the legs, but they have the flagellum or the hair-like structure, flagellum, with the help of which they will be moving. Then the next one is cytoplasm. Inside the bacteria, the jelly-like fluid which is present, that is called as the cytoplasm, where because of which the growth of the bacteria will be happening, the metabolism of the bacteria will be done through this. Then if they want to divide, they will be dividing in the cytoplasm. So all those growth, maintenance, metabolism will be done inside this fluid called as the cytoplasm. Then the next one are the plasmids. This structure is called as plasmid. Plasmid is basically like this. They are the DNA molecules. So though they do not have the well-developed DNA, but they have the little undeveloped form of it, which is called as a plasmid. So plasmid are small circular DNA molecules that are separated from the chromosomal DNA found in the bacteria. So whatever the DNA, they do the replication of the cell, the same way they will also be working on it. They will also replicate the same process in the body of the bacteria. Then the next part are the pink structures, which you can see, they are the nucleoid. So as I told you, they do not have the nucleus. Nucleus are not present in them, but they have the nucleoid which contains the DNA. Nucleoid is basically undeveloped form of the nucleus. It is like, sometimes when we go to some restaurant and all, what happened? They will be cooking the food. Sometimes those chapatis are not cooked properly, right? They will be uncooked. So in the body of the bacteria also, since they do not have the nucleus, they have undeveloped form of the nucleus, which is called as a nucleoid. And this nucleoid contains the DNA. The last one are the ribosomes. They will be helping to actually receive and to transfer the information of protein. So whatever the DNA has, the DNA characters have, they will be replicating along with the ribosome. So ribosome, they will be helping in the replication process of it, and because of which they will be passing the information to the other bacteria through the division of it. So these are the parts of the bacterial cell. Okay. I am just copying it again. Just a minute. Yashoda, can you read what is the function of the capsule?
The capsule is an outer layer that protects the bacteria. It also serves, I can't see. One sec. It serves several purposes, including helping the bacteria attach itself to the surface or other organisms as a form of defense and a nutrient reserve.
Okay. Okay. Hansini, read the function of the pilus.
Thin hair-like structures that protrude from the cell surface of many bacteria. Their main purpose is to assist the bacteria in attaching itself to a surface or to the other bacteria.
Okay. Sahiti, the function of the cell wall.
The main purpose of the cell wall is to maintain the shape of the bacteria and give it some protection.
Okay, good. Kirtana, the function of plasma membrane. Kirtana. Okay, Priyamvada, you read it. Function of plasma membrane.
Plasma membrane regulates the movement of substances in and out of the cell.
Yeah, tell me.
Mom, can you hear me?
Kirtana, yeah, I can hear you, Kirtana, now. So, Kirtana, read the function of the flagellum.
The flagellum assists with the movement of the bacteria by rotating much like a propeller.
Correct. Srivatsa, read the function of the cytoplasm.
Okay, the cytoplasm. The cytoplasm is where essential functions of cell growth, metabolism, or replication will occur.
Okay, read for the plasmid also.
So the plasmids are small circular DNA molecules that are separated from the chromosal DNA found in bacteria.
Okay, good. Charan, read the function of nucleoid and ribosome.
Okay, ma'am. Nuclear. This region in a bacteria cell contains most or all of the genetic material, DNA. Ribosome. The role of ribosomes in bacteria is to receive and translate genetic instructions for the formation of proteins.
Okay, good. Fine. So that's about the bacterial cell. So they can give you directly explain the bacterial cell, or they can ask you to write about the prokaryotic cell. In both the ways, the answer will remain the same. That is, you need to draw the bacteria diagram with the labeling of it and the functions of each and every part of it. You should write it. So after the class, I will be sharing this note. So make sure that you go through this diagram and also learn the function of each and every part of it. Now, moving forward to the next one is the differences between the nucleus and the nucleoid. Nucleus and the sorry, spelling of nucleoid. The first difference is that the nucleus, they are larger in size. They are larger in size. Whereas the nucleoid, they are comparatively smaller in size. So it is comparatively smaller in size. This is the first difference between the nucleus and nucleoid. Then the nucleus will have the covering. So they will have the outer covering, which has two membrane. So that is what we called as double membrane covering. So it has a covering of double membrane envelope. Like this. I'll just draw it. So suppose this is the nucleus. Outside the nucleus, they will have two more layers present around it. Whereas in the nucleoid, that membrane is absent, or that covering is absent. So a covering membrane is absent in them. The next one, nucleolus is present in them. So they have the small round spherical structure present in them. Whereas in them, it is absent. Nucleolus is absent. Then the next one, its DNA content is equal to two or several DNA molecules. So whatever the DNA they have, it is basically equal to many DNA molecules. So the functioning is so fast because of this nucleus only. Whereas in the nucleoid, they have the DNA whose content will be equal to a single DNA molecule. Here, single DNA molecule means a kind of the single chromosome functioning of it. Then the last point, its DNA is associated with histone protein. So they have a protein in them which is called as histone protein. And this histone protein, what do? They form the chromatin, chromatin. So histone protein, they will be forming the chromatin. Chromatin are what? They are the thread-like structure. And this chromatin will form the chromosome. So nucleus, in the nucleus, whatever the DNA is present, they will have the histone protein. The histone protein will make the chromatin and chromatin will make the chromosome. That is how they will be working. Whereas in the nucleoid, the histone are absent. So in it, the histone proteins are absent. Now, if they do not have the histone protein, can they form any kind of the layer around it? No, they cannot. So that is why the DNA of a nucleoid, so whatever the DNA the nucleoid has, they are often naked. It means they are not being covered because they do not have any kind of the histone protein. So they are naked. So these are the differences between the nucleus and nucleoid. Nucleus larger in size, the nucleoid they are smaller in size. Nucleus they have the double membrane envelope covering, whereas nucleoid they do not have that membrane. Whereas the nucleus they will have the nucleolus present in them. The nucleoid will not have the nucleolus. Then nucleus will have the DNA content which is equal to many several DNA molecules, whereas the nucleoid will have the DNA content which is equal to only one single DNA molecule. Then the nucleus will have the histone protein in them which help in the formation of the chromatin and the chromosome, whereas the nucleoid, in them the protein molecules are absent, because of which they are often naked. Am I clear, Yashoda and Srivatsa?
Yes, ma'am.
Srivatsa,
Yes, ma'am.
Sahiti, Priyamvada, Hansini, Kirtana,
Understood, ma'am.
Charan.
Understood, ma'am.
So these are like, 3 marks question directly they will be asking you in the exam. So just we'll have a quick kind of the revision. Prokaryotic and eukaryotic. Pro means old, primitive, or the false. And karyotic is a word derived from the karyon, which means the nucleus. So they will have the old nucleus or the primitive nucleus or the false nucleus. Or we can say in a very simple language that in them the nucleus is not clearly defined, or the nucleus is absent, or it is like, without any kind of the membrane. Whereas in the eukaryotic, the nucleus will be clearly defined because they have the advanced nucleus. Then coming to the differences, prokaryotic nucleus is absent, whereas in eukaryotic it is present. Prokaryotic, they will have nuclear membrane which is absent. Then prokaryotic, they have single cell, whereas membrane bound organelles are absent in them. In the eukaryotic, they are present. In the prokaryotic, cell division will take place by the fission or by the budding, whereas in the eukaryotic, two division will take place, which is the mitotic and the meiotic division. And in the prokaryotic, the size will be small, and the eukaryotic, the size will be the large in them. Then bacterial cell, different parts they have: capsule, cell wall, pilus, plasma membrane, flagellum, mesosomes, ribosomes, nucleoid, plasmids, and cytoplasm. Then the last what we have discussed are the nucleus and the nucleoid part of it. So any doubt any one of you have till here?
No.
No? Okay. So we have said, like, I have used this word many times, DNA, 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 but what it is DNA? DNA stands for the deoxyribonucleic acid, which is the genetic material. This DNA is what? The full form of DNA is deoxyribonucleic acid. It is a type of genetic material. What do they do? They actually help in transferring of characteristics from parents to offspring. So you are similar to your mother, or maybe you are similar to your father. Why it is so? It is because of the DNA. From your parents' body, that DNA has been copied and that DNA has been passed to your body. That is why might be your face is similar to your mother or maybe your father. You might be knowing it better, right? For me, in my case, I'm very much similar to my mother, maybe. So because of that reason, it is like the DNA, which gives the transformation of it, the transferring of the characteristics from the parents to the offspring. And that is the reason you might have seen in the movies and all, they will be saying, like, if they are not able to identify who is the real father, real mother of that particular child, so what they will be doing, they will be doing the DNA test, because the DNA of each and every individual is different. So like, if I have the phone, in the phone I'll be keeping the password. Most of us are keeping a different password, right? So a code will be there. When you have the laptop also, you will be setting some codes and all. So that password kind of the thing is also present in our body, which is your password or your code will be similar to your father and the mother. So that is how they identify through the DNA test whether that particular child belongs to that particular parent or not, through the DNA test or DNA copying or DNA fingerprinting also. Clear? So in the next class now, then we will be starting with the major part, which is about the nucleus structure, then the cytoplasm, the cell wall, and the cell organelles. Okay, children?
Yes.
Okay, ma'am.
Srivatsa, what do you mean by DNA?
Deoxyribonucleic acid.
Read the spelling. Spelling is important because it comes for the 1 mark.
For DNA, right?
Yeah,
So
DNA.
DEOXY.
Hmm.
So RIBONUCLEICACID.
Okay. So DNA, deoxyribonucleic acid. What it is? If you have to write the definition, what you will be writing?
So it's like the transferring of characteristics from parents to offspring.
That is a function. So if you have
That's
to
the
start the
function.
definition, you should start it like it is a genetic material which helps in transferring. Okay. It is a genetic material which helps in transferring of characteristics from parents to the offspring.
Okay, ma'am.
Hmm. Okay. So children, I'll be sharing these notes now. So make sure that you go through them once. Yesterday's notes and today's notes both together I'll be sending. Go through everything, read it, learn it by heart. Okay, then only we'll be moving to the next part in the next class. Otherwise, we won't be. Okay, children, then. Thank you.
Thank you, ma'am. Bye.
Thank you, Shrivasa.